The lab is sort of organized chaos. There are about 35 or 40 people in it, graduate students, postdoctoral fellows, technicians, support staff, etc. We have probably an unusually broad research program. We focus in three major areas. We work on the biology of small RNAs. We work on cancer biology, usually with focus on the roles of small RNAs in cancer and ways in which we can use small RNAs as a tool to understand cancer. And then we have sort of a third area, which is technology development and genomics, and mostly making use of some new generation sequencing technologies to try to understand everything from the evolution of cancer to human evolution. Well, RNA interference actually now describes a fairly broad range of biological phenomena. The notion is that RNA has a sequence just like DNA does, and that sequence can be used to recognize complementary RNAs that share sort of the inverse of that sequence, the mirror image of that sequence, if you want to think about it that way. And through that recognition, a lot of jobs can be done. The RNAs that are recognized can be destroyed, they can be taken to different places in the cell, or they can even guide processes as strange as taking pieces out of the genome, depending on what organism you're talking about. So one of the sort of evolutionarily deepest rooted roles of RNAi is as a genome defense. It's a way that plants, for example, recognize and fight viruses. It's a way that animals recognize parasitic pieces of DNA within their own genomes called transposons and shut those off. It's also a way that the cell uses RNA to program the regulation of its own genes, and we can exploit any one of these responses, essentially tricking the RNAi machinery into silencing any gene that we want, just by fooling the machinery into recognizing it as, in essence, one of these foreign elements. And we can use that for a number of purposes. The one that we mainly use it for is for trying to understand the biology of those genes. And again, in our case, mostly trying to understand what different sets of genes do in tumor development. Well, we've done a number of experiments in mice to try to figure out really what RNAi does in animals, and I can give you a couple of examples. So one is we've looked at the small RNAs that the cell makes in order to regulate its own genes and compared the spectrum of those small RNAs in normal cells versus tumor cells. In one of the first cases that we did this was in a tumor type called B-cell lymphoma. And by making that comparison, we discovered that there were a set of microRNAs, which are what these endogenous small RNAs are called, that differed between the normal cells and the tumor cells. And it turns out that the locus, the gene which encodes those microRNAs, is often amplified in that specific tumor type. And if we reproduce that event, adding extra copies of those microRNA genes, we actually accelerate the development of that particular tumor in mice. Another way that we approach this problem is by taking all of the components of the RNAi machinery, all the proteins that actually bind to the set of small RNAs and that the small RNAs program to do these regulatory events, and delete them from the genome and essentially ask what the consequences are. And it was in part those experiments that led us to the realization that RNAi in animals plays this sort of genome defensive role, protecting the DNA of germ cells from the ravages of these mobile genetic elements called transposons. Well, for a long time we've been interested in using RNAi as a tool to silence genes of interest, whatever genes we want. And in fact, my lab has built large collections of RNAi-inducing agents, in fact, collections that could be used to turn off every gene in the human and mouse genome. Now, the way that we use these to investigate cancer biology is essentially to take a set of cancer cells, engineer them so that each cell has a different gene turned off. And then we ask how those cells react under stress. What genes do cancer cells require that normal cells don't? These are potential targets. for therapy, what genes modify the response of cancer cells to chemotherapeutic agents or targeted therapies, what genes modify the ability of cancer cells to engraft in a host, to metastasize, etc. Well, there are really two ways that one would move from the work that we're doing into something that would be applicable in the clinic. One is a process that's being repeated in many pharmaceutical companies, many labs, really built on the foundation of understanding this basic piece of biology, which is searching for unique vulnerabilities in tumor cells. Now, once one finds a cell upon which a particular subtype of breast cancer, for example, depends, that the normal mammary epithelial cells don't depend on, then one can find targeted agents, using standard pharmaceutical chemistry, that could inhibit the activity of that particular target. And there you would have a potentially selective therapy in the way that things like Gleevec are selective for specific gene rearrangement upon which the tumor cells carrying that rearrangement uniquely depend. Another promise of RNAi is that it can be used as a therapy itself, and there is a tremendous interest in this both in the academic and industrial communities. The notion that pharmaceutical chemistry can only access about 20% of the genes encoded in the genome, because we have a limited ability to regulate chemically the activity of the proteins encoded by the other 80%. RNAi doesn't have such a limitation. All it needs to be able to do is to specifically recognize the sequence of that gene in order to shut it off. And so it is essentially, in some ways, a potentially universal pharmaceutical approach. The difficulty is, and I think this is where many of us are focusing a lot of effort, is trying to figure out how to take this relatively large molecule compared to a normal pharmaceutical and get it efficiently delivered to the cells in which the therapy needs to operate. And so really that is one of the major barriers to taking this basic science discovery and really exploiting it as a tool for improving human health. Well, I think that the way this barrier to delivery will be overcome is chemistry. So it's clear that we've pushed the biology of RNAi as a silencing tool sufficiently far that, given an ability to deliver this, we could shut off any gene that we wanted to. And in fact, we could probably dial the activity of these inhibitors to the point that we could even figure out exactly how far down we have to turn a gene. Maybe you don't want to shut it off because maybe it's essential. Maybe you want to inhibit its activity 50% or 80%. And the tools, our understanding of the biology is sufficient that we can achieve that with the tools that we have. I think that where we'll see advances in delivery come from is in changing the chemistry of the small RNA itself sufficiently that it can pass through cell membranes without the aid of a specific delivery agent. Delivery agents are the other sort of arm of research into RNAi delivery, encapsulating RNAs in lipids, in polymers, and that's another strategy for getting small RNAs into cells. But my own feeling is that the unformulated molecules probably have the best potential to be a future therapy. It's a simpler formulation than something that, where you're mixing an RNA agent and a series, for example, of polymers to try to get them across the cell. Well, this is, I think, an interesting story about the social aspects of science in a way. The realization that this transmissible cancer even existed came from a graduate student in my lab who happened to be Tasmanian. She did her PhD on sort of basic aspects of RNAi biology, and when she finished her degree, before she moved on to a position as a postdoctoral fellow, she was looking for a transition project. And she explained to me that this transmissible cancer existed, that in the last 10 years it had wiped out roughly half of the devil population, and that it was estimated to actually drive the devils to extinction sometime within the next 30 or so years if nothing were to be done. So she was very passionate about this and convinced me that we had the tools to try to understand at least something about the biology of this disease. Now, there are only 2 transmissible cancers that I know about where the cancer itself, the cancer cells themselves, move from one individual to another. There's this Tasmanian devil facial tumor, and there's also a tumor that does this in dogs. In dogs, the tumor isn't as aggressive. It often spontaneously goes into remission and doesn't often kill the animals that are in essence infected with this. In Tasmanian devils, the tumor is extremely aggressive, often kills affected animals in roughly 6 months with these horrific facial tumors that ultimately kill the animal, often because they grow so large that the animal can no longer eat. But it's a very aggressive disease also in the sense that it metastasizes to many sites in the body, but the animals are killed so quickly that it's usually not the metastases that cause them to succumb, as happens in many instances of human cancer. So our basic question was really to try to understand the origins of this transmissible tumor, to try to understand what cell type the tumor is derived from, so that we could maybe gain some insight into why this tumor has such an unusual etiology, and also to try to understand why it is that this tumor can spread from individual to individual, as that's such an unusual property. We came at it from a number of different perspectives. First, really founded upon our work in small RNAs. So each cell type expresses a specific repertoire of small RNAs that it uses to regulate genes, but that repertoire is also kind of a signature of the cell type that those small RNAs are expressed in. So you can use a small RNA signature to tell a mammary cell from a germ cell. You can tell a nerve cell from a muscle cell. And so we started off by asking what spectrum of small RNAs these devil facial tumors expressed. And that pointed us, in addition to looking at the messenger RNAs, the RNAs that encode the proteins within that cell, by looking at the small RNA profiles and the messenger RNA profiles, that really led us to a specific cell type, which in this case happens to be a Schwann cell. Now, what Schwann cells are is essentially like the insulation on a wire. They wrap nerve cells and act essentially as an insulator to make their conductance of signals more efficient. Well, I mean, I think that the 2 things that really come out of the work that we've done, looking both at the tumor and at the host animals, are that the host animals themselves are remarkably genetically homogeneous. And so one, I think, insight that emerged from the study was that the notion that the tumors may be able to spread from animal to animal, essentially like a metastasis, but within a population rather than within an organism, in part because the animals can't recognize these cells as foreign. Now, in terms of a treatment, it's difficult to envision wild animals undergoing cancer therapy. You can't really imagine a mobile ICU in the Tasmanian bush. But one thing that one can consider is a prophylactic therapy. And so cancer vaccines have kind of a bad rap, in part because they haven't worked particularly well for humans. But the unusual nature of the Tasmanian devil tumor, the fact that every tumor is identical, is a very different situation than one has with human cancers, where every cancer is different, is individual not only to the person, but in its genetic composition. And so in some cases, the difficulties of developing cancer vaccines in humans might be related to the heterogeneity of tumors, whereas one has precisely the opposite situation in Tasmanian devils. So one can really envision 2 approaches. One is the approach that's being taken, which is to take a set of devils and set them aside as an insurance population. And so you essentially sequester disease-free devils with the idea that, should the population actually go extinct in the wild, you have animals to reintroduce. Now, in that case, we can use the work that we've done, which in essence led to a diagnostic marker for this disease, to ensure that any tumors that insurance population might develop aren't DFTD. In the second instance, one might imagine trying to develop a vaccine against this particular unusual tumor cell that could potentially be used on wild populations. Although, again, you can imagine the challenges that one would face in doing that. Well, so although each cancer is unique, there are certain core pathways, biological modules in essence, that are altered in nearly every cancer cell. And the number of those modules expands as one gets more specific to a particular subtype, let's say estrogen receptor positive breast cancer. So the notion, and this is one of the ways in which I think RNAI is being deployed very powerfully, is to try to correlate the vulnerabilities that are created by the inactivation of those individual biological modules and then exploit that vulnerability for therapy. The way that you can think about this is that cells are designed or evolved to be robust. And every time you take away one of these biological modules, these pathways, it affects the robustness of cells. Think about a Jenga tower, and you're pulling blocks out of the Jenga tower. The more blocks that you pull out, the easier it is to make that tower fall. And what people are doing with RNAI is trying to figure out precisely what additional block to pull out to make that tower fall, where the tower wouldn't fall if one had a normal cell, which essentially has homeostatic mechanisms to be robust, to resist the biological difficulties that would be created by the loss of the pathway that would represent that key therapeutic target. And so the idea is to exploit common aspects of the genetics to create therapies that overcome the individuality of the disease. Well, so although each cancer is unique, there are certain core pathways, biological modules in essence, that are altered in nearly every cancer cell. And the number of those modules expands as one gets more specific to a particular subtype, let's say estrogen receptor positive breast cancer. So the notion, and this is one of the ways in which I think RNAI is being deployed very powerfully, is to try to correlate the vulnerabilities that are created by the inactivation of those individual biological modules and then exploit that vulnerability for therapy. The way that you can think about this is that cells are designed or evolved to be robust. And every time you take away one of these biological modules, these pathways, it affects the robustness of cells. Think about a Jenga tower, and you're pulling blocks out of the Jenga tower. The more blocks that you pull out, the easier it is to make that tower fall. And what people are doing with RNAI is trying to figure out precisely what additional block to pull out to make that tower fall, where the tower wouldn't fall if one had a normal cell, which essentially has homeostatic mechanisms to be robust, to resist the biological difficulties that would be created by the loss of the pathway that would represent that key therapeutic target. And so the idea is to exploit common aspects of the genetics to create therapies that overcome the individuality of the disease. You know, I came from a very small town. I had very good and very dedicated science teachers in high school who really went, far beyond the call in a way to make additional science training available after school hours, etc. doing things like science competitions that were held at local colleges, things like this, and even making available some very basic college science courses during our later years of high school. Going on to undergraduate, I went to Case Western Reserve, a private university in Cleveland, with the idea that I was probably going to do medicine. The first semester I was there, I needed a job, and talked to my undergraduate advisor, who said, "Hey, why don't you come work in my lab?" And it took about a week to decide that, that's what I really wanted to do. And so, for me, and I think for many, the turning point comes as an undergraduate student when you become exposed to the world of scientific research. It's not something that a lot of people are intimately familiar with. It's not a job that most people see or encounter. And so I think the key opportunity is to give undergraduates the chance to come into the lab, to provide them exposure to what a life in academic science is really like. Graduate education is something that at Cold Spring Harbor we've spent a lot of time thinking about, and really Jim Watson pushed very hard about a decade ago to encourage us to fight against the length of graduate training, which was increasing to what he felt were ridiculous time frames. And so, a 7-year PhD, a student essentially not becoming an independent scientist until well into their 30s was something that he thought completely inappropriate. And so we've really tried to essentially reinvent graduate education in a way that allows biologists to get a PhD degree in a short amount of time. 4 years is our goal. We tend to meet it by and large. I think over the 10 years that our graduate program has been running, our average is about 4 years and 3 months, and in fact, I graduated a student yesterday in 3.5 years. And so, the notion of bringing people into research early and then giving them an opportunity to do independent work while they're, young and full of fire is, I think, a really critical issue that the community is trying to address. Well, we work for the public. If you want to think about where the funding comes for our research, it comes from the National Institutes of Health, which is supported by taxpayers. And I think consequently we have a real obligation to communicate science to the public. Outreach, I think, should happen at every level. I think that working scientists should be interfacing with high schools, for example, going into high school biology classrooms, communicating the excitement of their work to students. Cold Spring Harbor Lab has a very strong community outreach program, something they call the DNA Learning Center, and it's designed to bring middle school students, high school students, into laboratories and expose them to laboratory science. I think it's our obligation as scientists to reach out to the public in the form of public lecture series. We also have a strong public lecture program at the laboratory. I participate in it. I know that many of the other investigators at the lab participate in it. And I think that the public has to understand science, not only because they pay for it, but because science is increasingly a part of their everyday life. And to go back to the discussion of sequencing and bioethics, a time will come in the not too distant future when you walk into a doctor's office and they sequence at least part of your genome. And without a basic understanding of biology, how is the general public going to react to this? How are they going to understand what's being communicated to them about their risk of disease and about how their own genetics affect their response to treatments? Well, we work for the public. If you want to think about where the funding comes for our research, it comes from the National Institutes of Health, which is supported by taxpayers. And I think consequently we have a real obligation to communicate science to the public. Outreach, I think, should happen at every level. I think that working scientists should be interfacing with high schools, for example, going into high school biology classrooms, communicating the excitement of their work to students. Cold Spring Harbor Lab has a very strong community outreach program, something they call the DNA Learning Center, and it's designed to bring middle school students, high school students, into laboratories and expose them to laboratory science. I think it's our obligation as scientists to reach out to the public in the form of public lecture series. We also have a strong public lecture program at the laboratory. I participated in it. I know that many of the other investigators at the lab participate in it. And I think that the public has to understand science, not only because they pay for it, but because science is increasingly a part of their everyday life. And to go back to the discussion of sequencing and bioethics, a time will come in the not too distant future when you walk into a doctor's office and they sequence at least part of your genome. And without a basic understanding of biology, how is the general public going to react to this? How are they going to understand what's being communicated to them about their risk of disease and about how their own genetics affect their response to treatments? Personally, to me, what's exciting about research is the moment of discovery. I think for most scientists, it's what addicts you to this. The idea that for just a few minutes you know something that nobody else in the world knows. And it doesn't happen often, because science is really sort of an exercise in banging your head against the wall over and over again. Scientists need a very high tolerance for failure and frustration, because most experiments do fail. But when something really works and you really learn something fundamentally new, it's a feeling that I think I've never experienced in any other way.